Hello, everyone. I'm Karina Xavier from the Gulbenkian Institute. So in our group, we are interested in understanding how cell-to-cell communication in bacteria, such as signaling process like quorum sensing, regulate bacterial group behaviors in complex multi-species bacterial communities. In particular, we are interested in understanding how microbe-microbe interactions take place in the community of symbionts residing in the mammalian gut, and how these processes, such as cell-to-cell communication and microbe-microbe interactions, play a role in the assembly, maintenance, and restoring of this multi-species bacterial community. So I think you all know that this mammalian, these symbionts that reside in the mammalian gut are important for many aspects of host health. And this, for example, include the production of lytic enzymes that are produced by the members of the gut microbiota, which are very important for nutrition because they can, for example, degrade, they are essential for degrading fibers from plants, which the host cannot digest. So they do have an important role in nutrition. Microbiota members from the gut can produce essential compounds like vitamin. They have a very important role in the maturation of the immune system. And they have a very important role also in the colonization resistance capacity of the microbiota. In my laboratory, we are particularly interested in this property of the microbiota, which is the colonization resistance, which is the ability that a healthy microbiota community has to act as a protective barrier against the invasion of pathogens or the expansion of pathobionts. And the reason why I'm particularly focusing on this function of the microbiota is because it's here where I think microbe-microbe interactions will play a more direct role. But we also think by understanding the molecular mechanisms that are involved in the colonization resistance property of the microbiota, we'll be able to identify important functions, molecular functions of the microbiota, which are then likely to also play a role in the other processes where the microbiota can influence host health and physiology. Given my previous knowledge in studying bacterial signaling and my understanding and our understanding that there are signal molecules called quorum sensing molecules, which are exemplified here in this cartoon, that are well known to regulate gene expression and group behaviors in bacteria, we want to know that if this, we want to know if these signaling mechanisms are also playing a role in influencing the behavior of the microbiota and the assembly of the gut microbiota. So these quorum sensing signaling molecules, which many of them are produced by the gut members, they regulate the process that, as I'm saying, it's called quorum sensing. And what we know from bacterial cultures in the laboratory is that many bacteria are capable of producing these molecules that we call autoinducers, which at low cell density are released to the extracellular media, but given the low cell density and the low concentration of the molecules, they are not detected by the bacteria. But when the bacteria reach a population size that it's at high cell density, the concentration of these molecules will increase proportionally, and it will then be detected by the bacteria, changing the gene expression profile of the population and allow bacteria to synchronize their gene expression and act and engage in group behaviors. So typically what we see in quorum sensing regulated processes is that quorum sensing regulates processes that are only productive at high cell density and are not productive at low cell density. So this is what we've learned from in vitro laboratory studies, and also as examples of bacterial group behaviors. We know that quorum sensing can regulate bacterial group behaviors such as production of bioluminescence, production of lytic enzymes like proteases or sugar polysaccharides degrading enzymes. And they are also involved in regulating processes important for biofilm formation, which are all group behaviors that are only productive at high cell density. And this type of behaviors have been studied very well in these organisms, but also in many others. And they've been shown to be important for both symbiosis and pathogenesis. But this type of symbiosis that have been studied before are usually very specialized symbiosis, as in the case of the vibrio and the squid symbiosis and the light production that is regulated by quorum sensing. We have here an example of Pseudomonas aeruginosa, which is a human pathogen that regulates biofilm formation and proteases through quorum sensing, and it's and this is important during the colonization of the lungs by in cystic fibrosis patients. Then there's another example here of a plant pathogen that regulates the production of plant cell wall degrading enzymes that can cause maceration in plant tissues. Here I have some potatoes infected with a wild type Pectobacterium strain or a quorum sensing mutant that cannot macerate the tissue. So to sum up, what I'm saying is that bacterial quorum sensing can regulate bacterial group behaviors, which are important, which have been shown to be important for specialized symbiosis and for pathogenesis. So I think that many of these behaviors that I just showed you are likely to be important also for the symbionts colonizing the mammalian gut. And these are things like production of biofilms, which have not been very addressed in the context of the gut microbiota, but I think they will be important. And also the production of the lytic enzymes, which are extracellular enzymes that I alluded to in the beginning when talking about the importance of these enzymes in degrading plant fibers that the host cannot degrade. So, thinking that maybe these processes are regulated by quorum sensing, we started to look at the function of quorum sensing molecules by studying this molecule here, which is called autoinducer 2. I chose to study this molecule in particular because the synthase for autoinducer 2, which is called Lexes, it's widely conserved among the bacterial kingdom, and it's produced by many members of the microbiota. And it regulates, importantly, it's known to foster a communication between bacteria across species. So it's the only molecule that we know can foster interspecies quorum sensing across very distant phylogenetic distant bacteria. So we established in our lab, we established a mouse model to be able to manipulate the levels of this autoinducer molecule, autoinducer 2 molecule, so that we can manipulate the levels of this signal directly in the mouse gut. So the mouse model that we started up with is a model where we use where streptomycin induces dysbiosis. So we're using antibiotic-induced dysbiosis that causes a huge change in the microbiota composition. So here we have a treatment of streptomycin of 28 days. And when before treatment, in the composition of the mouse microbiota is composed typically by firmicutes and bacteroidetes, which is a very common composition found in the mammalian gut, where these two major phyla are usually quite well balanced in a approximately 1-to-1 ratio. And what happens upon this long streptomycin treatment is that the firmicutes are severely impaired and the bacteroidetes dominate. So the streptomycin-induced dysbiosis is characterized by the domination of the bacteroidetes. And at the functional level, what we observe when we treat the mice with streptomycin is that before treatment, the levels of these bacteria, Bacteroides vulgatus, are very low. But after treatment, we see this expansion of the bacteroidetes, and it's mainly the main cause of this domination is characterized by a huge expansion of Bacteroides vulgatus, which has been shown to cause inflammation under certain conditions. So it's usually called a pathobiont. And the other thing that happens is that if we challenge mice before treatment with E. coli cannot invade and cannot colonize the mouse gut. But if the mice are first treated with streptomycin for 2 days, then we see a loss of colonization resistance, and E. coli can colonize the mouse at very high levels. So what happens in streptomycin is that we see pathobiont expansion and loss of colonization resistance against pathogens. Here I'm illustrating this with E. coli, but this is also true for Salmonella, for example. So we use this model to ask if quorum sensing could be influencing the microbiotic composition during the antibiotic treatment and affecting the outcomes of dysbiosis. So to manipulate the autoinducer 2 levels in the mouse gut, we took advantage of a system that I had discovered in E. coli, that E. coli is capable of both producing but as well as degrading the autoinducer 2 levels in vitro. So we constructed several engineered E. coli strains that we then tested in vitro and confirmed that they accumulate different levels of AI2 activity in the media. And then we colonized mice with no E. coli or different E. coli mutants. And we could see that both the wild type and this mutant that is being constructed to not degrade the signal can accumulate AI2 levels to very high levels. And this strain can actually decrease the natural AI2 levels present in the gut. So then what my students did was to look at the microbiotic composition after 28 days of streptomycin treatment in 3 different groups of mice that were colonized with three different E. coli engineered strains. So there was a group of mice that was colonized with an E. coli that induces high levels or produces high levels of autoinducer 2, an E. coli that can degrade and remove autoinducer 2 from the gut, and a control strain that neither produces or degrades the autoinducer 2. And then this, I'm going quite fast in this part because this has been published, but what we observed in these 3 different groups of mice, when we looked at the phylum level, at the highest phylogenetic level, at the microbiota composition after 28 days of treatment with streptomycin, was that the mice that had been colonized with the E. coli that can accumulate high levels of AI2 had higher levals of firmicutes than the other 2 groups. And on the other hand, they had lower levels of Bacteroidetes. So increasing autoinducer 2 by E. coli led to changes on the microbiota composition at the phylum level, and high AI2 favored the expansion of firmicutes while hindering the expansion of Bacteroidetes in this antibiotic-induced dysbiosis. And so to summarize in a cartoon manner, this a long story short, and as I said, this has been published and you can look at all the other experiments that we did. But what the take-home message from this part is that in an untreated microbiotic composition, we have a composition that is usually found in the mammalian gut with a good proportion between Bacteroidetes and the firmicutes. This proportion between these 2 phyla is important for many functions of the microbiota. Upon streptomycin, we see a dominance of the firmicutes. And what happened in our experiment is that the E. coli producing AI2 was able to favor the firmicutes, and therefore it counteracted the negative effect of the streptomycin. So high AI2 favored the emergence of firmicutes in the antibiotic-treated mice, countering the dysbiosis induced by the long-term antibiotic treatment. Okay, so now from now on, what I'm showing has not been published yet, and it's really the follow-up of this first study that we did, where we had established this mouse model to manipulate the autoinducer 2 in the gut, and we studied its consequences in antibiotic-induced dysbiosis. So now we want to understand the molecular basis behind this response to AI2 during antibiotic-induced dysbiosis, and more specifically, we wanted to identify microbe-microbe interactions that were taking place during this process, and that would change in the presence of autoindustry 2. So to identify the important microbe interactions taking place in our experimental setup, we teamed up, my student Rita Oliveira teamed up with Karina Xavier from Sloan Kettering and applied the model that he had used before. It's an ecological-based model, mathematical model, based on the extended Lotte-Volterra equations that basically we use bacterial abundance of the different species, the most abundant species during our experiment to model the time course dynamics of microbiota changes. And then we can look at what happens during a perturbation and identify which are the stronger interactions and upon this streptomycin perturbation, but also the effect of the E. coli and the E. coli that is producing autoindustry 2, and look for interactions that would be changing according to autoindustry 2. So in this experiment, what Rita did was to use the two conditions where we had seen the strongest changes, which were having, again, the streptomycin treatment and then looking at E. coli, the control strain that doesn't manipulate the levels of autoindustry 2, or the E. coli strain that increases the levels of this current sensing signal. And then we also introduced another group where we had no E. coli. And so then we took time course microbiota data from fecal samples and then applied the Lotte-Volterra model. And the first thing that we could observe was that there's an expansion of Bacteroides vulgatus, as I had alluded to before. And this expansion of Bacteroides vulgatus during streptomycin perturbation was much stronger when there was no E. coli than when there was the E. coli producing AI2. So the streptomycin treatment favors the expansion of B. vulgatus, but this expansion is less pronounced in the presence of the AI2, and so E. coli seems to be inhibiting B. vulgatus expansion. And then we looked for the interactions that could be the most relevant interactions during all these three conditions, inferred by the model. And we saw a very interesting interaction between B. vulgatus and B. ovatus. So what happens between Bacteroides vulgatus and Bacteroides ovatus is that when there's no E. coli, we see a positive interaction between these two organisms. It seems that ovatus is helping vulgatus. According to the model, it seems that ovatus is helping vulgatus to expand in the streptomycin-treated gut. And when we introduce E. coli, this interaction becomes slightly negative, but is highly negative in the presence of the E. coli producing AI2. So it seems that this expansion of vulgatus is inhibited according to the model, is inhibited by AI2, and that the model predicts that there's a positive interaction a cooperative interaction between the two, and that AI2 might inhibit this cooperation. And we thought this was interesting because, as I mentioned before, vulgatus seems to be a phytobiont, and there was already a report of a possible cooperation between these two organisms. So the model predicts an easy testable hypothesis that vulgatus is inhibited in the presence of AI2, and that this cooperation or this interaction, which becomes negative here, is regulated by AI2. Okay, so then we reasoned that maybe this interaction between vulgatus and ovatus could be a response to AI2 changing the gene expression of these organisms. And to test this hypothesis, we used a more simplified model, which would allow us to do transcriptomics, where we use a germ-free mice that we colonize with the defined bacterial cocktail, where we have the genome sequence of all these bacteria. They are 12 bacteria, which are highly, where bacteroidetes are highly abundant, similarly to our streptomycin model. And this defined bacteria cocktail also included Ovatus and Vulgatus. So then we did RNA-seq from mice that were colonized with this defined bacterial cocktail that were previously either colonized with the E. coli control strain or the E. coli control or the E. coli strain that increases the autoinducer 2 levels. And then, as you can see here, there were several genes that were differentially expressed in these two groups, either in Vulgatus and Ovatus, meaning that there were genes that were differently expressed in these two organisms in the group that either had the E. coli producing AI2 or the E. coli control strain. So now we focus in two particular operons that we thought could give us some light in this potential cooperative interaction between Ovatus and Vulgatus. And these operons were highly significantly differently expressed, being in Vulgatus here and this Ovatus here. So the expression was higher under low AI2 environment. So the expression of these operons is possibly being inhibited by autoinducer 2. And the reason why we got interested in these operons is that they are part of the SAS operons, the starch utilizations, the operons that are involved in oligosaccharides utilization, and they are related with the metabolism of fucosylated or sialidated oligosaccharides. In Vulgatus, we saw a fucosidase and a sialidase, and in Ovatus, we saw a fucose permease. And the data indicates that this would be, that AI2 might be inhibiting fucose metabolism in B. Ovatus and B. Vulgatus. So what is the source of these fucosidated oligosaccharides and sialidated oligosaccharides? So what happens is host glycan is typically protected by fucosyl moieties, and most of the times the members of the microbiota cannot have access to the glycans from the host because of these fucose molecules that protect the glycans. Only a few bacteria from the microbiota can access these host glycans. But according to the genome sequence, B. vulgatus do have a fucosidase. So this means that most likely they are capable of removing these fucose molecules and then degrade host glycan. And what we think might be happening is that vulgatus can remove these moieties here, and then when it does so, they both can then share the glycans, and actually then B. vulgatus can use the fucose, the free fucose released during the degradation. And this is what we could predict from the genome. And in laboratory cultures, we could see that indeed B. vulgatus can grow in a fucosylated oligosaccharide or fucosylated saccharide, and the Ovatus cannot. And in fucose, Ovatus seems to be better at growing than vulgatus. So then we also did co-cultures, and these co-cultures, although I'm simplifying here and removing some of the controls, I think these co-cultures indicate there's indeed a possible cooperative interaction between these organisms. So in this case, we have here in Bivu ovatus cannot grow into this fucosylated oligosaccharide. And we used inulin as a complex saccharide that we knew Ovatus could grow here in black, but vulgatus cannot. And then vulgatus, as I mentioned before, can grow in the fucosylated compound, but we see the best growth when we have the two bacteria growing together in the two compounds. So if you compare the black with this yellow curve, what you can see is that we have the fastest growth rate when the two bacteria are together using the compounds that only one can use. So this is an indication that they can cooperate, and at least in the test tube. So now going back to the AI2 regulation, as I said, there's a fucosidase, and this fucosidase, the gene name is 2433, that according to the RNA-seq data in vivo, seem to be repressed in the presence of high AI2. So we constructed a promoter fusion to this gene and then tested in vitro cultures, and we can see we constructed a mutant that cannot produce the autoinducer 2 signal, and we can see that vulgatus expression of this fucosidase is higher when there's no autoinducer 2, and we can supplement the mutant that is impaired in autoinducer 2 synthesis with exogenous AI2 and observe this repression. So this data suggests that indeed autoinducer 2 can repress the expression of the fucosidase. And then we looked for potential receptors for sensing autoinducer 2 in vulgatus, and we found this putative receptor called 166 that has structural homology to an AI2 receptor, and we purified the enzyme, and it seems to bind autoinducer 2. And in the mutant that doesn't have the receptor and that cannot produce AI2, and then when we add back AI2, we don't see changes in gene expression of the fucosidase. So this regulation was abolished when we removed the receptor. So it seems that indeed vulgatus can regulate, autoinducer 2 can repress fucosidase, and it has a sensor that is involved in this regulation. Okay, so this is actually where we are at the moment. And now we want to do the same thing in Bacteroides vulgatus, where we have to construct a mutant in the FreeCos permease and study its regulation, which is what I think is missing to then validate our current working model, where we are proposing that vulgatus and novatus are cooperating when the autoinducer 2 levels are low, but by inhibiting the fucosidase and possibly the permease in these two organisms, autoinducer 2 will inhibit this cooperation. So to summarize what I've showed you, in a streptomycin-induced dysbiosis, we see an expansion of Bacteroides, in particular B. vulgatus, and this expansion is inhibited by AI2. So autoinducer 2 seems to counteract the dysbiosis, the antibiotic-induced dysbiosis. And what we think is the major driver of this expansion is this cooperation of vulgatus with ovatus, and that the fucosylated the ability of B. vulgatus to degrade fucosylated mucus glycan is important for this cooperation and expansion. And we are testing the model where we think AI2 inhibits this gene expression between these two organisms, inhibiting the cooperation between the two. So to sum up, AI2 manipulation led to changes in gene expression that we have already observed in vitro in vulgatus, confirming the RNA-seq results in vivo. And this, and we think this is important for the counteracting the antibiotic-induced dysbiosis that we observed in the streptomycin model, therefore inhibiting pathobiont expansion. So this is my thank you slide with the members of current members of my group, and most of the data that I showed was done by Rita Oliveira, who's just finishing up to move to a postdoc in the US next month, and we're set to see her go, but very happy for her. Thank you all for listening, and I'm free to answer questions.
Thank you, Karina. It was really nice talk. Any questions? So I'll start with the general question for the AI2 in our species. So could you do something like this in lactobacillus and give it to the people that actually have problems in the gut dysbiosis because inflammatory bowel disease or something like that. Would that help with because they also have high levels of bacteroids, I think.
So sorry, I didn't listen to the first part, but you're asking if we could restore dysbiosis by giving lactobacillus, right?
That would produce more AA2, the same way that you did with E. coli. Could you do the same in this species?
Okay, yeah, lactobacillus indeed can produce AI2, and it is one of the probiotics. So yes, maybe. So we are indeed looking at other ways of manipulating autoindustry 2, as opposed to the one that we thought was the easiest, which was with the E. coli, that we can engineer, and we knew which conditions we had to give to synchronization. But definitely, lactobacillus could be a way. I think we would be able to increase the levels, but not to decrease the levels. But meanwhile, we have other strains like this commensal Klebsiella that we identified recently, which can also be used to manipulate autoindustry 2. And I think we're going to start testing that one first. But yeah.
Okay. Olibek is asking, would you expect Vulgatus Chauvatus AA2 interplay be impaired in the FUT2 conditional knockout mice? Not sure what is this mice.
He said it's in the FUC2. You're saying in the probably it's this mouse knockout that doesn't have that the glycans are not fucosylated, and that's something we would like to test. If indeed our experiments are correct that the fucose molecules are in the glycans are important. Maybe this cooperation will not be present in the FUC2 mouse knockout. Yeah, I think that's a good hint. We had thought about that, but we don't have the mice.
Barbara asks if the AA2 in the gut fluctuates during the day and if it changes during the fasting. If you have after feeding, low feeding.
Yes, we have not done that because in order to get at the moment still to get good data on AI2 activity, we do have to sacrifice the mice, so we have not done like time course experiments, but we are still working in having a good sensor that we can see in more real time, and that would be interesting to see. Yes.
And also, Lebek is asking, could other species like Akkermansia or Ruminococcus also contribute as well to this interplay? Yes. So in the model was able to infer interactions between that were influencing Vulgatus between 4 bacteria, and that included Ruminococcus. Under these conditions, Akkermansia was quite low. So we didn't see a signal for Akkermansia, but we do have, so we usually see Akkermansia, for example, when we use Gnotto models, when we reintroduce the microbiota there, so to germ-free mice. And we have not yet seen a response that really popped out for us related to Akkermansia, but Ruminococcus for sure, yes. Okay, no more questions. Thank you, Karina. It was really nice. Thank you all and thank our speakers for these 2 amazing talks. And if again we are telling if someone doesn't have to be PhD students or postdocs would like to speak with the speakers, please send an email, and we will see you all in a month.